Thank you for kind introduction and thank you for attending for this session. And so as an introduction, I'm Toshi, a postdoc in Wendell Lim Lab at UCSF. And today I'd like to briefly present what we are doing, how we can apply synthetic biology approach to like a development, especially stem cell based embryo model. So at least right now, there are two major approaches to construct like in vitro model. One is simply adding a signal directly to the media. Most of organoid or like a gastroid rely on this approach. So, but because we simply add like a growth factors or drug to the media, that means that all the progenitor cell or ESL receive a signal and pushing them to the specific fate. So that means like most of the organoid, uh, uh, focus on like a specific organ or tissue. And in case of gastroid, they are focusing on like a posterior side of the gastration. In contrast, like Magda or Yakub Hana lab recently developed a new approach and it is widely known as like a synthetic embryo. What they did is that they mix the ESL with the extra embryonic cell and add extra embryonic stem cells locally secrete the morphogen so that like ESL aggregate or epiblast have like a compressed morphogen gradient. And so that in their approach, they can like reconstruct like almost entire embryo from like a head, heart, and like a somite to tail. But I think limitation of this approach is that because the genetic circuit of extra embryonic cells are highly complex, so mechanisms are usually black box, and we cannot control timing and the level of the morphogen induction. So what we want to propose here is that to introduce a new cell-based approach that can generate morphogen gradient with user-defined orientation, amplitude, and the signal type. So inspired by the classic organizer concept, we developed a synthetic version that can assemble into a defined structure and provide a spatially precise morphogen. And so far, we have developed two different types of the structure. One we now call like a node that can act as a localized source of the morphogen. And the second one is we are calling a shell that covers the surface of the target ES aggregate and provide morphogen uniformly. So to build such node and shell structure, we engineered the cell adhesion of the synthetic organizer cell. And we use a mouse fibroblast cell line for the base cell line for the synthetic organizer cell, because wild type mouse fibroblast cell line doesn't have any impact on the ESL. So to introduce some heterotypic interaction between mouse ESL and our fibroblast cell line, we introduce the synthetic adhesion molecule, the SYNCAM, which is developed in our lab. So now the ESL engineers to express the non-fluorescent GFP on its surface, and those non-fluorescent GFP are recognized by anti-GFP expressed on the synthetic organizer cell. And because now we have like a physical interaction between GFP and anti-GFP, now synthetic organizer cell, so like fibroblast cell can attach on the surface of the ESL aggregate. In contrast, if you overexpress certain type of the cadherin, such as a PCAD, the PCAD can introduce a homotypic interaction between the cell, so that now the fibroblast cell can form a cluster. And to ensure that the node is attached on the specific point of the ESL aggregate, now we also added the SYNCAM on the to the node. And the right panels are actual images of the synthetic organizer cell. So the center is the cluster of the ESL, and the colored cells are synthetic organizer cell. And as you see that now we can make this kind of single cell layer around the ESL aggregate. we are calling shell, and the node that is a cluster directly attached fused to the ESL aggregate. And we can simply combine these form together to make a shell plus node or like even two node structure. And once we build the node and shell structure, we next focus on how we can add temporal control over morphogen signaling in the synthetic organizer system. So to turning on the signal, we simply introduce a DOX inducible system. So now when the node receives the DOX, they start to secrete the Wnt or target morphogen, and the expression level monitored by BFP signal. And to turn off the signal, we use this ICAPS9 suicide switch-based system, which triggers the apoptosis signal. So when the node receives the drug, now within the 24 hours, the node becomes dead, which is judged by that the node is detached from the target ESL aggregate, and also we don't see any BFP signal inside the node. And we can easily remove this kind of dead node by simply changing the media. And so we next try to examine whether our synthetic organizer cell can secrete morphogen and activate the downstream cascade inside the ESL. And we first focus on the Wnt signaling pathway, especially Wnt3A, which is a key morphogen that specifies the posterior side of the embryo. And to monitor Wnt activity in the ESL, we use this PTCF emission signal so now when the cell receives the Wnt, it starts expressing the MHA. So uniform activation of the Wnt through the Wnt3A in media or using Wnt3A secreting shell. Now you see the uniform activation of the MHA reporter around day 4. But in contrast, if you use a Wnt3A node, now you see the local activation making a gradient inside the target ESL aggregate. And also Wnt is widely known as like a growth factor. And we also detected like elongation towards the Wnt3A node. So both results suggest that clearly that Wnt3A node can drive the symmetry breaking event. But the true power of using the synthetic organizer system is that now we can systematically control the shape of the Wnt gradient. So as I mentioned in last slide, so we can make a gradient using the Wnt3A node, but the gradient the steepness is quite shallow. That means that the cell really far from the Wnt3A node also activated the Wnt signal. But you can like turn on or like make the gradient steeper by introducing the inhibitory signal, such as like a DKK1 node. And in this case, now you don't see any activity close to the DKK1 node, making the overall gradient steeper. And you can make the gradient even steeper by covering its surface with a DKK1 shell. And now you see that there's only local Wnt activity right next to the Wnt3A node. And of course, in extreme case, like with only DKK1 node, you don't see any Wnt activation. And what is a critical thing is that those difference of the gradient, the steepness of gradient, will determine the cell distribution, especially the patterning of anterior and posterior cell lineages. So we first checked the germ cell patterning around day 4 and found that the Wnt3A node induced a BRCA positive cell around the Wnt3A node, but less like a SOX2 positive cell. So it means that it is inducing like a primitive streak state. But in case of the dual node cases, so both Wnt3A node and DKK1 node cases, we do observe both like BRCA positive cell around the Wnt3A node and the SOX2 positive cell closer to the DKK1 node. In these cases, so we are getting like both primitive streak and the ectodermal cell. And in case of the DKK1 node only cases, there's the most of the cells are SOX2 positive, so it is shifting to like ectodermal phase. And when you continue culturing a little bit more longer, like we found an interesting phenotype emerges around day 8. And that is that the local cardiac beating domain emerges on the opposite side of the Winsley node, which is examined by the immunostaining of cardiac troponin. But when you add the DKK1 node factor, because DKK1 node make the gradient steeper, it pushes the cardiac domain towards the Winsley node. And now in this case, you see that the cardiac domain emerges in the middle of the body. And instead, like OTX2 positive anterior neuronal fate emerges around the DKK1 node. And on only DKK1 node cases, you don't see any cardiomyocyte, but still you can get like anterior neural fate around the DKK1 node. So quick conclusion is that if you want to have both like mesodermal fate and your neuronal fate, you need both like you need a right balance of like activator and the inhibitor in the system. But that doesn't mean that, the simple Windsor node cases are useless. And it is actually still quite interesting because still you can get like a local cardiac beating domain in case of the Windsor node, which is quite different from like when you compare with like normal cardiac organoid. So cardiac organoid usually depends on like adding a lot of growth factor in the media, and you will get this kind of like cardiomyocyte everywhere. But probably because we limited the source of the morphogen, we always get like a local bleeding phenotype. So we decided to analyze a little bit more detail about this phenomena. So first, we try to characterize how this kind of local bleeding phenotype emerges by using this KDR reporter line. That's kind of then KDR is the progenitor of a marker for the progenitor of vascular endothelial cell. And as you see that, so first around day 5, you will get like a KDR positive cell around the center of the body, but it gradually shifted to the opposite side of the Windsor node. And eventually around day 8, you do see this kind of beating cardiac chamber emerges. And also outside the beating chamber, looks like there's a vascular network is emerging. And we further confirm by immunostaining of cardiac troponin and PE-CAML, and we confirm that indeed that cardiomyocyte, so cardiac troponin positive cardiomyocyte surround the beating chamber, and and also like in the main body, it looks like angiogenesis or like vascular network is emerging. So the quick conclusion of this talk is that so we developed a synthetic organizer that have spatial control of spatial and temporal control over the morphogen signal. And synthetic organizers can systematically prove the relationship between signaling gradient and the resulting morphological outcome. So I skipped quite a lot of stuff, but you can read the full story in this paper. And also, I'm planning to apply on the job market this cycle. So if there's a good position, please let me know. And this is a acknowledgement site. So I really thank my PIs, Wendell and also Ophir, who guides the project and also mentoring me. And I also thank Benoit. He helped us quite a lot on the cardiac part. And of course, a lot of people are involved in this and help the project. And I don't have time to thank all the people, but I want to thank Coralie. She is a co-founder and she was a very great collaborator. And also I thank Dario. I think I skipped all the data of the single cell analysis, but in paper there's a lot of the data. And Dario did all the single cell analysis, so I really thank the both of them. So that's all for my talk, and thanks for listening, and I'm happy to take a question.
Yeah, thank you so much for the amazing work. Yeah, if you have questions, please type them in the Q&A box. We have a question here from Alex. In your Win3A slide, it looks like the Win3A stays around the same size, whereas the neural node increases in volume. So does the node cells proliferate, and does their cargo affect this cell behavior?
Yeah, that's a good question. So as we are using like a living cell, it actually so the synthetic organizer cell also grow. But what we did a small trick is that so we pick a single cell clone that grows really slow. And it looks like when you have the cell line with higher expression of the PCAD usually grow slow. So there's like anti-correlation between like a PCAD expression and slow growth. So we are picking the high express PCAD that is at the same time it grow really slow to, keep the size relatively same over like the time course of the experiment. Um, so that's a trick. And I do think that the size matters for the final phenotype. We haven't analyzed like systematically, and we try to keep the node on the same size. Um, but I think that the controlling the size of like controlling the size and the growth of the synthetic organelle cell gonna be probably the next step.
Okay, yeah, great. So we have another question from Dagon Seagull. Sorry if I pronounced the name wrong. Very cool talk. Naive question. For the two-node model, can you
Mhm.
spatially control that they will be on opposite ends?
Yeah, so that's the biggest, I think, technical limitation right now. And we highlighted that we cannot control the relative position in our supplemental figure. So it's really, they just randomly attach on the surface of the ES target ES aggregate. So we are just picking up the two nodes that uh, position opposite side. Um, Yeah, well, we are thinking some kind of solution, but yeah, that's also kind of like a future walk.
I see. We have one more question from Ranan. Your images and videos are impressive. Could you briefly summarize how you label the whole mount organoids?
Mm. So I think it's um just follow normal procedures, and so we just fix. So we are using the 96 well U bottom plate. So I did all the staining like fixation and like blocking in the 96 well U bottom plate. And also, so we did the tissue clearing using the cubic, and that's all about the procedure. Nothing special, I think. Well, just use the tissue clearing, and we do need to image with a laser scan confocal, not like a spinning disk, to get like a of higher resolution. There's some a detailed protocol in the paper, and if you have a further questions, just email me. I can give you more details.
Okay, great. Okay, one last question. Um, Sorry, actually the same question, another question from Renan.
Mhm.
They're also curious what type of extracellular matrix you're using.
Oh, we are not using ECM or extracellular matrix. No Metrogel. At this work, we can put the Metrogel and that we are doing actually right now, and it looks like adding Metrogel will globally shift the phenotype. But at least the data I presented today, there's no like Metrogel.
Okay, one last quick question. Can you comment on the development of definite endoderm in your system?
Yeah, so as we first focus on like a Wnt signaling pathway, so there's almost no endodermal state. So we run the single cell RNA-seq and we confirm that there's a very few, maybe less than 1% of the endodermal cell. But that's probably because we focus on the Wnt. And we also use the activin A node which is in supplemental figure, I think, in the paper. And in that case, I think the cell mostly shifted to endodermal fate, which is confirmed by, I think, FOXA2 and SOX17 positive immunostaining.
Okay, thank you so much, Toshi, for your great work.